Hello students, welcome to EPG Pathshala. My name is Dr. Priyanka and I teach Anthropology at the Department of Anthropology, Dr. Hari Singh Gaur University, Sagar. Today we are going to discuss about the module on genetic load and mutation under the paper Human Population Genetics. So before we proceed towards this module, let's see what we are going to learn from this module. In this module, we will be covering the concept of mutation and its various types. We will be understanding about the concept of genetic load. We will also see that what are the various factors contributing to genetic load in our population. Molecular anthropology is a field by which goals of anthropology are served using techniques and methods of molecular biology. Molecular anthropology is a relatively young field of research. It has developed both methods and theories and have extended its application from covering areas of human evolution such as reconstruction of the history of human population and peopling processes, the characterization of DNA in extinct humans, and the role of adaptive processes in shaping the genetic diversity of our species. Molecular anthropological studies help us to determine evolutionary link between the ancient and modern human population as well as contemporary species by examining the DNA sequence in different populations. This information help us in understanding the closeness or the disparities between population at genomic level. Besides tracing the migration and settlement pattern and genetic proximity and disparities, molecular anthropology also help us in understanding the health or the disease profile of a population. It helps us in deciphering the molecular basis of adaptation in a population. Research in the field of molecular genetic studies includes broadly the human evolutionary genetic studies, biomedical studies, and primate genetic studies. As we all know, human genome is not static and it is subjected to a variety of heritable changes. These changes are important as without the occurrence of hereditary changes, life form could not have been evolved and diversified. Alternate form of a gene can be resulted due to the sudden heritable change in the sequence of the DNA. Mutation is defined as the abrupt change in the genotype or the gene of an organism that is not the result of recombination. Recombination, on the other hand, is a process of mutual exchange of segments between homologous chromosomes. Recombination is a process of reshuffling of alleles. But as such, recombination do not give rise to the new alleles. It only gives rise to the new combination of alleles. Mutation, on the other hand, is a process which gives rise to the new alleles. Mutation includes all types of heritable changes of an organism. The process by which mutation is introduced in a gene is termed as mutagenesis, whereas the resultant phenotype with mutation is called as mutant. The factor which leads to the mutation is called as mutagen. Mutation is the ultimate source of genetic variation. It provides the raw material for evolution to occur. It results from the formation of an allele where new organism will be able to evolve and adapt to the environmental change. Thus, mutation is used to designate process by which hereditary changes arise. There are several characteristics features of mutations, such as mutations are generally recessive, but dominant mutation also occurs. Mutation is a random process and it can occur at any time in any cell of the organism. Mutation can be recurrent, as the same mutation can occur again and again. Mutation can occur at any stage of development. If it occurs at the germinal cell stage, we call as a mutation occurring at germinal cell stage. Mutation in germinal cells are basically very important, as the germinal cells are undifferentiated cells which give rise to gametes. So, the germinal cell mutation influences several gametes and further influences all the individual derived from them. Mutation in case of gametes or zygote also occur. In this case, gamete is affected and therefore the individual will be the only one who will be carrying this mutation, not all the gametes. Mutation in a cell. In this stage, the mutation occurs after the zygote has undergone one or more division, and therefore only a part of a body will show a mutant characteristics. The mutation at this stage is called as somatic mutation. Mutation can be of various types. They are classified into different categories based on various criteria. These criteria can be method of detection, agents of their cause, or the level at which they occur. The different types of mutations are morphological mutation, lethal mutation, biochemical mutation, resistant mutation, spontaneous mutation, induced mutation, point mutation, and conditional mutation. We will learn about each one of them one by one. Let's see what exactly are morphological mutation. Morphological mutation affects the outward appearance of an individual, including color, shape, size, etc. For example, occurrence of albinism in man is such an example of morphological mutation. Lethal mutation. Lethal mutations are also known as deleterious mutation. They are any sudden change in the genetic material leading to the death of the individual. Next in the row is a biochemical mutation. These type of mutations are identified by a deficiency of a particular protein or enzyme or nucleic acid. Enzyme, as you all know, are nothing but protein itself. For example, disorder of G6PD is the example of biochemical mutation. G6PD disorder is caused due to the deficiency of glucose 6-phosphate dehydrogenase enzyme. Next is a resistant mutation. This type of mutation is identified by their ability to grow in the presence of a pathogen or antibiotic. As you all know, resistance to particular drug is an example of this type of mutation. Next is a conditional mutation. Conditional mutation is a type of mutation which allows the mutant phenotype to express under permissive conditions. Now let's come towards another kind of a mutation known as point mutation. A point mutation causes replacement of a single base nucleotide with another base nucleotide in the genetic material. Depending upon the effect of this replacement, point mutations can be classified into three types: missense mutation, nonsense mutation, and silent mutation. Missense mutation is a type of a mutation that alters the codon and introduces a different amino acid. Nonsense mutation, on the other hand, is a type of a mutation where sequence of DNA results in a premature stop codon. That means no further messenger RNA molecule will be synthesized. This results in the formation of incomplete amino acid chain or faulty protein. Silent mutation is a kind of a mutation which results in no change of amino acid or its functionality when the altered or changed messenger RNA is translated. Let's revise what is the meaning of a codon since we are using it again and again. Codon is a triplet of nucleotide base specifying a particular amino acid. For example, codon TTC code for lysine amino acid. The figure given in this slide basically shows the three kinds of a point mutation. As we have discussed, there are three types of point mutation: silent, nonsense, missense. We'll begin with a situation when no mutations has occurred. For example, here at DNA level, we have a codon of TTC which code for protein lysine through messenger RNA. In case of a silent protein point mutation, code TTC gets converted into TTT. Though there is a change in a DNA base, but there is no corresponding change in the amino acid. In case of a nonsense point mutation, the change from TTC has occurred to ATC. This change has resulted into no amino acid formation in the protein chain. The next one is a missense mutation. In case of missense mutation, at the DNA level where the nucleotide base sequence is TTC can convert to TCC or TGC. In both the cases, a wrong amino acid is inserted into the protein chain, forming a faulty protein. Depending upon the cause of mutation, mutation can be of two types. They can be spontaneous mutation or they can be induced mutations. Spontaneous mutations are the type of mutation that occurs without treatment of organism with an exogenous mutagen. Mutagens are the substance which results in mutation. Spontaneous mutation is the ultimate source of natural genetic variation in populations. Spontaneous mutation arise from a variety of sources, including error in DNA replication, transposition of transposable genetic elements, and spontaneous lesions. Induced mutation, on the other hand, are the type of mutation which arises due to the presence of an exogenous mutagen. It is a reaction with the DNA, which results in structural change that affects the base pairing capabilities of the altered nucleotide. Mutations can be artificially induced with the help of two broad categories of mutagens: physical mutagens and chemical mutagens. Physical mutagens are basically the radiations such as UV radiation, X-rays, etc. The effect of mutagen depends on its wavelength and penetrance. Lower is the wavelength, higher the penetrance, and more the damage to the DNA. Chemical mutagens, on the other hand, are chemicals that induce mutations. Chemicals used for inducing mutations are mustard gas, ethyl urethane, formaldehyde, and phenol. Chemicals acting as mutagens was first detected during World War II. Watson and Crick proposed the structure of DNA. According to them, the structure of DNA is quite stable, but there are still chances of some alteration in the DNA. Each gene is a potential site for a mutation. Yet, some genes mutate more frequently than the other genes. We call this region where more frequently mutation can occur as the mutational hotspots. Mutation at the molecular level have a permanent alteration in the sequence of nucleotide in the nucleic acid. Structural mutation are of two types. One is a gene mutation and another is a chromosomal structural mutation. A chemical change that occurs in the DNA of a cell is called as a gene mutation or a point mutation. Such mutation may alter the sequence of a nucleotide within a part of a DNA molecule. This alternation changes the information on the DNA chain, as a result of which wrong messenger RNA is synthesized and because of which wrong protein is synthesized. If the DNA undergo replication, Because of this process, wrong DNA will be replicated. Tautomeric shift is one such example of point mutation. In this, pairing quality of the bases are changed. Usually, purines pair with pyrimidine, but in this case, purine pairs with purine and so on. Tautomerism is caused by certain chemical mutagen. There are other categories of gene mutations, such as transition, transversion, and frame shift. Transition are substitution gene mutation in which a purine is replaced by a purine or a vice versa. Transversion, on the other hand, is a kind of a mutation where a purine is replaced by pyrimidine. Base pair addition and deletion are collectively known as frame shift mutations because they alter the reading frame of all the base pair triplets. The other type of a mutation that occur at the chromosomal level are deletion, translocation, insertion, inversion, and duplication. In case of a deletion, loss of a piece of a chromosome happens. Duplication occurs when one extra but identical piece of a chromosome is added to the normal chromosome. When an inversion occurs, the piece of a chromosome breaks apart and pieces rejoin the same chromosome but in a different order. Translocation is a process by which a portion of one chromosome is transferred to another chromosome. In case of insertion, a portion of one chromosome has been deleted from its normal place and reinserted into the another chromosome. In this diagram, we can understand the mutations at molecular level. The first figure shows the deletion of a segment of a DNA. In this figure, we can see a segment of DNA being deleted, resulting in the short size of the chromosome. The deletion results into the loss of the genes also. In case of a translocation, a segment of a chromosomes get translocated to another chromosome. In case of duplication mutation, a segment of the DNA or the genes also gets duplicated in number. In case of inversion, a segment of the DNA breaks and is inverted and again rejoins in an inverse manner. We can also understand the same principle by seeing the DNA sequences bases. In this picture, we see A, B, C, D, E, F, G is the original sequence of bases in the chromosome. In case duplication happens, a particular DNA base, in this picture D, gets double in amount. In case of a deletion, D gets omitted from the DNA sequence. In case of an inversion, the sequence get inverted. Like in this case, the sequence called as DEF get placed as FED. It can happen in any segment of the DNA. So let's revise once again the two important type of mutations, point mutation and frame shift mutation. In case of a point mutation, there is a change of a single nucleotide base. This change can result into deletion, insertion, or substitution of one nucleotide in a gene. In case of a frame shift mutation, a base pair addition or deletion happens. This addition or deletion changes the entire frame of reading the codon. Now, let's move to biochemical level from molecular level. As we all know, genes control various traits in an organism by controlling synthesis of proteins and expressing themselves through synthesis of enzymes. Beadle and Totem proposed one such hypothesis for the action of enzymes. which is one gene, one hypothesis. The hypothesis states that if a biosynthetic pathway, several steps are involved, each step will be controlled by a specific enzyme, which is synthesized under the control of a specific gene. Inborn error of metabolism in man are the best example showing the effect of mutation at biochemical pathways. Inborn error of metabolism are a group of disorders which are genetically transmitted and causes a single gene defect by blocking significantly the metabolic pathway. There are three important diseases associated with inborn error of metabolism. They are phenylketonuria, L-captonuria, and albinism. In phenylketonuria, accumulation of phenylpyruvic acid causes mental retardation. Children suffering from phenylketonuria are called phenylpyruvic idiots and are thus unable to break phenylpyruvic acid into hydroxyl phenylpyruvic acid. In case of L-captonuria, accumulation of homogentisic acid happens. The urine of the patients suffering from this disease turns black when it comes in contact with the air. In humans, albinism is a result of a mutation in the pathway which converts dihydroxyl phenylalanine into melanin. Albinism is due to the absence of melanin pigment, and individuals suffering with this disease are incapable of converting this particular chemical into melanin. Now let's move towards the genetic load. The concept of genetic load was given by Helden and Mueller. It hovers around an idea that the genotype in a population have a mixture where one genotype is better than the another. Genetic load is the difference between the fitness of an expected optimal genotype with the fitness of observed average genotype in a population. It accompanies with a loss of a proportion of individual known as genetic death, as there is a probability of an organism failing to reach the reproductive age. This fact is important to study by scientists as the population with too high genetic load will be in danger of going extinct as the organisms are unable to survive and reproduce viable offsprings in the next generation. Types of genetic load. The first one is a mutational load. Genetic loads are affected by mutation which are negative, as more mutation in the genome are deleterious than being advantageous. In Crow terms, mutational load can be defined as the extent to which a population is impaired by recurrent mutation. Mutational load is larger in big populations with positive selection because there are more individuals that can be potential carrier of deleterious genes and can pass on the next generation. In positive selection, genome undergoes changes and also resorts the mutation which is beneficial to the population. The mutational load has two components, one produced by the deleterious mutation and the other produced by the beneficial mutations. Both the components of mutational load arises because an ideal population consists of individuals homozygous for both beneficial as well as deleterious genes. The next in the category is a segregational load. The segregational load is very simple as it is defined as a balancing load which comes into the play when heterozygote superiority arises in a population. Now let's see what are the different causes of genetic loads. The first can be inbreeding. Inbreeding increases the rate of homozygosity, and inbreeding depression will be observed when the offspring receives two copies and deleterious alleles, resulting in a lower fitness of the individual. A declining population will experience high rate of mutation. An increase in the rate of mutation in alleles will increase the pair frequency alleles. Due to decline in the population because of higher mutation rate, there is a chance of mating between the remaining organism to breed with their relatives, and thus it will help in pushing out some of the harmful mutations. Sexual reproduction also acts as a causing agent of increased genetic load. Sexual reproduction has a direct effect on the genetic load. It increases by pairing together of two different deleterious genes referred to the concealed portion of the genetic load. Sexual reproduction can also prevent the spread and maintenance of deleterious genes in a population due to elimination of less fit individuals. Genetic load will increase at a steady rate if mutations spreading to all offsprings are slightly deleterious to the point where an individual can survive and reproduce. Migration. Migration load results when an organism non-adaptive to a particular environment comes in contact with individual adapted to that particular environment. The offspring produced as a result of such mating are not as fit as they would have been if both the parents were adapted to the same environment. The offspring will have a lower variability rate as well as a lower overall fitness, and higher will be the genetic load. So let's summarize what we have covered in this module. In this module, we have understood that mutation can be rare, they can be lethal, randomly occurring, they can be recessive to dominant, reoccurring, that means they can occur many times, and they can happen at any time and span of the individual's lifetime. Mutation can occur at any stage in any cell. They can occur in germinal cell. They can also happen in gametic or zygotic cell stage. They can also take place in any cell of the body called as somatic cell. Mutation can be classed into different categories such as morphological mutation, lethal mutation, biochemical mutation, resistant mutation, spontaneous mutations, induced mutations, point mutation, and chromosomal aberrations. Mutations can be induced. That means they can be caused by application of some physical measure. Certain chemicals and radiation act as a potent mutagen that can cause mutation. You must have heard the example of X-rays and UV rays in causing mutations. Likewise, you also remember the very famous Bhopal gas tragedy, which causes mutation because of some chemical agent. Genetic load. Genetic load is the decrease in the fitness of average individual in a population relative to the fittest genotype due to the presence of deleterious genes in the gene pool. That means presence of deleterious or lethal gene in the gene pool can reduce the fitness of the individual bearing it. There can be many factors contributing to the genetic load in the population. These factors include factors such as inbreeding and gene flow. Thank you.